Good morning, everybody. So, some previous classes are completed in the part 1 of nutrition chapter. So, today is a chapter in the final topic of the part 1 in the nutrition, that is mechanism of photosynthesis. The mechanism of photosynthesis. So, already you know that everybody, what is photosynthesis? So, plants can prepare the food by using of sunlight and observing of some raw materials. The process is called here photosynthesis. Already you know that. But how to utilizing these raw materials? The why to we can how to use in the which mechanism involving are they here? So water we can observe the roots. How to we can process passing into the leaves? When enter the leaves in the water, how to we can splitting are there? After splitting, how to prepare in the carbon dioxide to convert a carbohydrate formation? These all we can different types of questions arise of in your mind. The questions in the clarifying the total in the process, the mechanism of photosynthesis. The mechanism means how the plants can prepare the food by explained by the that means explained by some diagrammatically or experimentally involving of the which mechanism are there and which process are there. So that's the topics today, that mechanism of photosynthesis. Let us see in the mechanism of photosynthesis. Mechanism of photosynthesis is divided into the two major phases are the here. Two major phases. One is called here light reaction, another one is called for dark reaction. In the mechanism of photosynthesis wise, two mechanisms are there. In the presence of light is light dependent reaction or light reaction. In absence of light is called for dark reaction or light independent reaction. In the mechanism of photosynthesis involving of the two basic steps are there, depend upon the reaction. Two basic steps, one is a light reaction, another one is dark reaction. This light reaction is light dependent reaction. That means this reaction that takes place of only present of light. And the light dependent independent reaction or dark reaction, this reaction does not depend upon the light. When absent of light, and this reaction we can, this mechanism we can start it. So, in the mechanism of photosynthesis, the total measure of two steps are there. One is a light reaction, another one is a dark reaction. In the first one is a light reaction. The first one is light reaction. In the light reaction, also called as photochemical phase. What is another name? Photochemical phase. The light reaction also called as photochemical phase or light dependent reaction also called. And also this reaction also called as yes the Hill reaction, because this reaction discovered by the scientist is Robert Hill. That's why this reaction name is called here Hill reaction. Is it clear? What is another name of in the light reaction? Photochemical phase, light dependent reaction, and also called as Hill reaction. Why they mention Hill reaction? Because in this reaction that discovered by the scientist name Robert Hill. That's why the mention is called for Hill reaction only here. So in the light reaction, light plays a important role. Yes or no? Yes, the lights plays important role. Here a series of some chemical reaction that take place one after another initiated by lights. Here total involving of only the light is a major key player. So where did this reaction we can occur? Some questions. Sir, in the light reaction that takes place in grana part. That means in the grana consisting of thylakoids are there. The light reaction takes place in the stacked saccharide structures are there. This plate is called for grana. The light reaction takes place in grana. In exact place, in thylakoid membranes are there here where the chlorophyll is presented. That place we can choose in the light reaction wise. So the light reaction that takes place in the grana of thylakoid membrane in present of chlorophyll. Present in chlorophyll. So already I'm saying the grana part here. That is the grana part here we can observe. In this place in the possible of only light reaction. So light reactions, some involving some steps are the here. What is the step one? We can observe here the sunlight in the falls the plants some type of in the rays the falling are the here. Here involving three base simple steps are the here. In the step 1, plant utilizing the sunlight, the solar energy. So when the smallest unit of energy is called photons. What is the smallest unit of energy here? Photons. So plants we can absorb the sunlight by using in the which wise only? Photons wise. The photon is the energy of the sunlight. You can see. So in the step 1, plant we can absorb the sunlight by using of energy wise, that is a photons wise only. Here is photons wise. So when the photons wise the absorb the energy, the what happened is that here the which process here. Here, in the first step, the reaction takes place in grana, a group of thylakoids which contains quantosomes. Because in the plant, we can trapping the sunlight with helping of only quantosomes are the here. Where the quantosomes are presented, that you can easily trapped by the sunlight. In the step 1, the plant trapped by the sunlight. In the step 1, how we can trapping are the here? Because in the chlorophyll pigment consisting of here some quantosomes are there. These quantosome we can trapping of sunlight, the energy store into the leaf part. How do we can energy store in here? Photons by storing of the energy. So these energy we can involving of the step 2 in the reaction is called for in the photolysis reaction. So the energy is used to split into the water molecule here. Okay, what happened is that here the water molecule split in the step 2. We can observe H2O in the gives as H plus and OH minus wise we can splitting. In the second step, when the energy enter in the leaf pod in the photosynthesis wise, then when the water transporting are the one place to another, automatic water molecules are splitting. How we can splitting are there? We can H plus and OH minus wise. So these H plus and OH minus we can splitting by using of only light. So that's why this reaction is called for photolysis reaction. We can observe. A photo means a light. Lysis means breaking. Involving of light, the breaking of water molecule. That's why this reaction is called photolysis. How the water we can divide? H plus and OH minus wise divide. Is it clear? But here by using of only H plus and OH minus and releasing of some electrons also. Not only H plus OH minus, when the water molecule are breaking time, the releasing of some electrons also. These electrons are very important in the step 3. We can observe. The highly reactive ions of water in the split into different direction as OH minus and H plus at the step 3. The OH minus ions through a series of in the steps produce the water and oxygen. So the oxygen we can release into the atmosphere in the process of atmosphere, but height H plus and we can continuous moving are the heres in the dark reaction involving are the heres. Is it clear? Can observe. What is the step 3? The step 1 is clear. Step 1 is energy entering the plants. The step 2 is the energy utilizing breakdown of the water molecule by converted H plus and OH minus ions to releasing of some electrons. And the third step, the H2 H plus and OH minus ions, the OH minus ions involving into the OH minus ions involving in the light reaction, the H plus ions involving into the dark reaction. So the OH minus ion, a continuous reaction takes place. The forming of water and oxygen are there. When the water utilizing the plants, oxygen we can releasing out of the plant body, we can atmosphere we can releasing out there. So when the H plus and OH minus and also releasing of some electrons also. So one electron or the first electron, the captured by the chlorophyll A. Chlorophyll A. So when the releasing of the first electron, the captured by the chlorophyll A is a process is forms primary photosynthetic reaction or photophosphor reaction is there here. When the electron became the captured by the chlorophyll A is which forms a primary photosynthesis or in the primary photophosphor reaction. That means in the electrons in the combine of chlorophyll A with formation of inorganic phosphate ion formation. That is IP. These inorganic phosphate ions we can absorb by the ADP converter into the ATP. and formation of NADP. You can clear. So when the electrons continues to absorb by the chlorophyll A to formation of inorganic phosphate ions, these inorganic phosphate ions of the energy converted from ADP to ATP and formation of NADP. These two ATP and NADP is called for assimilatory powers. What is the name here? Assimilatory powers. So these assimilatory powers by using into the dark reaction only. In the light reaction wise, when the present of light time, the using of sunlight, splitting of water molecule, releasing of electrons, the electron formation of the some assimilatory powers is the concept. What is the mechanism of light photosynthesis? Step 1 is using of sunlight energy by the photons wise, and the step 2 is water molecule within the splitting by the reaction of photolysis, and the step 3 is the formation of the electron converted here ATP and NADP asymmetry powers wise. So finally in the light reaction, 12 ATPs and 18 NADPH forms. Finally how many ATPs are there? 12 ATP and 18 NADPH forms. So what is the full form of ATP? Adenosine triphosphate is the full form. NADP means nicotine amide dinucleotide hydrogen phosphate. Once again I repeat, nicotine amide dinucleotide hydrogen phosphate is the NADPH. So in the light reaction, total how many ATPs and NADP form? 12 ATPs and 18 NADPs form. So this light reaction discovered by the Husserl Emerson Anderson Hill reaction, but exactly we can discover by the Emerson scientist. The scientist name Emerson. Okay, so this is one of the light reaction. We can observe the reaction here. What is the period? Adenosine dinucleotide hydrogen NADPH forming this called assimilatory powers. So how we can form a assimilatory powers? Electron ions to capture the chlorophyll A, formation of inorganic phosphate ion. These inorganic phosphate are converted ADP to ATP. This. So NADPH and ATP is called for assimilatory powers. And the second step is dark reaction. What is the second step? Dark. Is it clear, everybody? Light reaction. And the next dark reaction. Dark reaction also called as like independent reaction or biosynthetic reaction. The two names. The two names are the one is dark reaction or light independent reaction and biosynthetic reaction. So this reaction discovered by the Blackman scientists. What a good name scientist. So what is the name? Blackman, the black sheep. In the light reaction discovered here, Emerson, and the dark reaction discovered here, Blackman scientist. And so in this reaction, what happened is by using of assimilatory powers to preparing of the starch formation. Because there is no light, there is no absent of light night time here, plants what doing are the here to prepare the food. How do we can mechanism? A three steps are there we can here, releasing of one carbon dioxide are there that we can use it. So that these involving of only Melvin Kelvin cycle by utilizing are the NADPH and ATP powers. Because the dark reaction in the cycle wise only Kelvin cycle we can explain only here. During the dark phase reaction, the hydrogen of the NADPH combines with the carbon dioxide with help of ATP energy in order to produce glucose. So already using of NADPH are the here, the formation of glucose are the here. So first how we can convert it here, some intermediate compounds are the here. And say, so the first, this reaction takes place in stroma part. In the light reaction takes place in grana, but this reaction takes place in the which part only? Stroma, the liquid fluid part. And in the plant we can observe the gases were already containing a carbon dioxide is there. These carbon dioxide in combined with some of the substances there inside the plant that is called for RUBP. You are not BP, the name is RUBP, that means ribulose bisphosphate. What is the name RUBP? Ribulose, here observe, RUBP means ribulose bisphosphate. So this reaction occurs in the number of steps by using some small special intermediate compound. So carbon dioxide, we can observe some intermediate compound RUBP, that's ribulose bis phosphate, in the presence of some enzyme that is called for Rubisco enzyme. Rubisco enzyme. Present of enzyme here, Rubisco enzyme. To convert it here, phosphoglyceric acid was converted. Is it clear? In the first step, by using the assimilatory powers, but not using the assimilatory powers in this time. When the carbon dioxide absorbing on the RGBP product on intermediate compound in the presence of Rubisco enzyme, how to be converted in the phosphoglyceric acid. So in the water is splitted, light we can utilizing whatever the gases why the carbon dioxide absorb. Carbon dioxide enter the leaf to combine with one of the intermediate compound that is RUBP in the presence of enzyme Rubisco enzyme to convert it, how to convert it, phosphoglyceric acid. This phosphoglyceric acid here can observe the phosphoglyceric acid, the 5 carbon sugar molecule. I want the C6 means 6 carbon, but it's only 5 carbon sugar molecule. So when the phosphoglyceric acid combined, these phosphoglyceric acid utilizing of the assimilatory powers. Phosphoglyceric acid to observe the NADPH and place ATP we can observing to convert it here to glyceraldehyde phosphate to GAP wise we can convert it. The phosphoglyceric acid to absorb by the some asymmetric powers of NADPH and ATP to convert, how to convert it to glyceraldehyde phosphate. So 2 glyceraldehyde phosphate divided here, 1 Ga plus 1 Ga we can convert it. 1 glyceraldehyde phosphate with fix with stroma and form starch. What happened? 1 glyceraldehyde phosphate, the fix with stroma part and to form a starch formation, to glucose to convert to starch formation. Another glyceride phosphate is here. These another glyceride phosphate to combine with RUBP. When the cycle formation, that's when the Melvin Kelvin cycle are there here. So one cycle formation was. So another one glyceride phosphate combined with RUBP, ribulose 1,5 bis phosphate converted here RUBP wise. See, one glyceraldehyde phosphate combines with ribulose 1,5 bisphosphate to converted RUBP. These RUBP catching of carbon dioxide to converted here phosphoglyceric acid. These phosphate are divided one glyceride phosphate and one glyceride phosphate. This one gets converted is starch formation. So there are the 3 different steps involving into the dark reaction. That means different steps. You can clearly observe here in the dark reactions. What happened is a carbon dioxide converted here carbohydrate formation. In the light reaction, solar energy converted chemical energy, the chemical energy utilizing splitting of water molecule. So what is the main function of chloroplast structure wise? 3 function: the solar energy converted chemical energy, the splitting of water molecule, and reduction of carbon dioxide to converted carbohydrates. Is it clear in the mechanism wise? In the dark reaction, the utilizing of carbon dioxide converted in the carbohydrate formation. Remaining 2 steps are there. Let us see, I'm already saying how to begin the dark reaction process we can occur. So normally dark reaction by using of how to convert it carbon dioxide to observe RUBP, the below bisphosphate with presence of Rubisco enzyme converted here phosphoglyceric acid formation, that is PGA. The 6,3 phosphog in this phosphoglyceric acid I'm already saying by utilizing of assimilatory powers in the step 2. ATP in the one step and the second step is the power of NADPH observing are the here. So ATP and NADPH are this phosphoglyceric acid converted here. 2 glyceraldehyde phosphate wise we can convert it. One is the G3P glyceraldehyde phosphate. So remaining 5 glyceraldehyde phosphate are the here. These 5 glyceraldehyde phosphate converted here starch RUBP product we can releasing are there and regeneration of RUBP. But these 1 glyceraldehyde phosphate are there here. This we can convert it here starch formation. This is one reaction we can take place on the reaction. You can observe the similarities and dissimilarities of the light reaction and dark reaction. We can here releasing of assimilatory powers, but here we can utilizing of the assimilatory powers. Is going to occurs in the light present of light. Here the only absent of light. So this is one of the most important in the mechanism of photosynthesis. Is it clear, everybody? Or any doubts are there here? So I think there is no doubts in these places. So light reaction takes place in the grana, dark reaction takes place in the stroma. So it's the grana part and this is stroma when the total liquid position are there here. So in the these all are in the photosynthetic pigments. These all are in the photosynthetic pigments. It's a present of chlorophyll A and chlorophyll B carotenoids in the phycobilans are there here. Is it clear? So we can observe the one of the flowchart of the light reaction. The chlorophyll grana captures the light energy. How we can the captures are there here light energy? By using of photons wise. So when we capture the light energy, we can just split a water molecule out between H plus and OH minus ions, and the OH ions losses electrons to OH group wise converted. The OH combines formation of water and oxygen. These both are released in atmosphere, and remaining H plus and involving of the dark reaction. That means exit of electron from using ATP. Chlorophyll we can remove, hydrogen atoms we can remove. The reduction of power of only light reaction, and this is called here dark reaction that takes place often the stroma involving of only carbon dioxide, because when the carbon dioxide and the catalyze a photosynthetic enzyme from glucose plus water we can releasing when the condensed in the starch formation stored as the starch grains. We get remaining steps here. A clear egg, right? Carbon dioxide combined with RuBP in the presence of Rubisco converted phosphoglyceric acid is the first step, and the second step phosphoglyceric acid NADP plus ATP converted formation of one glyceraldehyde phosphate, one glycerol, two glycerol, one glyceraldehyde and another glyceraldehyde, one glyceraldehyde converted in the RuB plus 1,5 bis phosphate, and this one here.